Okay, thank you very much. I know I'm the last person between you and lunch, so I'll make sure this is worth your time. So my name is Bo Xiang. I'm an assistant professor at the National University of Singapore. And today I'll be talking about single-cell splicing and SQL analysis with ISSAC. So most of the work that I'll be talking about today is done by this very talented PhD student, Yuntian Zhang, from my group. So for those of you who are not familiar, splicing QTLs are naturally occurring variants that changes the inclusion and exclusion of exons. So they lead to different isoforms like such. And it's widely known that splicing and splicing QTL regulation is cell type specific. They occur in certain cells, but stronger in certain other cells. So a number of high-profile publications have studied single-cell QTLs. So you see that these papers, but all of them focuses on the expression effect or the genetic regulation of expression, which is eQTLs. And there's also a number of literature on splicing QTL analysis, but most of them, or actually all of them, are focusing on bulk splicing QTL. You'll be familiar with a lot of them. So for example, sQTL Seeker, sQTL Seeker 2, Dustal, Leafcutter, and Glimpse. However, single-cell splicing QTL mapping remains a major challenge. So we sort of tackled this challenge head-on from 2023 to 2024. So what we did is that we sampled 474 individuals of East Asian, Southeast Asian, and South Asian. These are the three prevailing populations in Singapore, which covers about 94% of the genetic diversity of Asia. Sampled about 1 million PBMCs. And then back then, we didn't have a very good single-cell splicing analysis pipeline, so we instead adapted Leafcutter, which was the de facto bulk sQTL analysis pipeline at the time. So what we did is that we used pseudobulk analysis, and then using Leafcutter, we were able to recover a complete pathway based on our single-cell splicing data, basically from regulatory variants to their eQTL, which changes the protein expression, which then changes the exon inclusion and exclusion of exon 4, 5, and 6, and eventually changes the cell type proportion of CD4 T cells, just using single-cell QTL data. And also we were able to co-localize our single-cell sQTL signal with GWAS signal and able to identify this risk gene of TCHP, and then we were able to validate its genetic effect using mini-gene assays. So we published this paper as the cover article of Nature Genetics, but this paper left us unsatisfied because of several reasons. The first reason is that there's no way to really map cell state dependent splicing QTLs. And this is because pseudobulk analysis by default is an average of all the cells in that cell type. So we basically average out all the cell state dependent QTLs. And the second is that there is a lot of single cell specific sequencing artifacts that are different from bulk sequencing artifacts, right? So this includes high PCR duplicates because we start with so low amount of RNA starting material. We have to do 10 more cycles of PCR, which significantly and exponentially increase the noise. And also the 5' and 3' read bias, which I'm showing here. So as you can see, this is the TSS, this red dotted line, and this is the TES, this red dotted line. So this is 5' 10x data, right? And you can see there is a severe bias towards the 5' end. And I'll come back to this effects later. So then we decided to improve on the tool. So we made this new tool called ISSAC, which stands for Integrative Single Cell Splicing Analysis and sQTL Caller. So I'll go through how this tool works step by step. The first step is that we're not doing pseudobulk anymore. So we were very ambitious initially. We wanted to do truly single cell, but then we were unable to do so because single cell is just too sparse for us to analyze any splicing. So instead, we settled on a sweet spot, which is meta cells. So basically, we aggregate somewhere between 10 to 20 cells per meta cell. So this reduces the sparsity significantly while allowing us to still get at cell type and cell state dependent QTLs. So, but this leads to a different problem, which is that, as you can see, if we use the standard QTL mapping, this is the phenotype Y, this is the genotype X, and this is covariates. So you see that there's repeated measures of the meta cells, right? So instead of one measurement per individual, now per individual you have many different measurements, right? So, so to mitigate this, we added a genetic relationship matrix in a mixed model. So what we do is that then we can model now the relationship between these meta cells, and we can properly account for their inter-individual relationships. So, and the second part, which is the single cell sequencing artifacts. So for the PCR duplicate, this one is easier to address, right? So this one, we can use the cell barcode and UMI-based recollapsing. So here, just a demonstration. For these two reads, they have the same cell barcodes and UMI, so we can collapse them. And also for these two reads, we can collapse them. And then instead of using Leafcutter's intron usage ratio, we devised this new novel usage ratio, which is what we call splice site usage ratio. So the first step is that we first define the splice sites. So here the font is a bit small, so I'm going to read them out: A, B, C, D, E, and F. So there's 6 different splice sites. And then suppose that we're interested in splice site C. The first step we do is that we get the junctions that includes splice site C. So in this case, it's this junction and this junction. And then we map to E, which is the splice sites that does not include junction C, but could have included junction C. So for example, we know that junction C could map to link to junction D and junction F. And then junction F instead have linked to another junction, junction A. So these are the exclusion reads. And then there's also the non-competitive junctions, right? So for example, between A to B and between E to F, these are non-competitive junctions. But these reads are there, but we're not considering them. So we define the splice site usage ratio as the reads that involve site C versus the reads that involves and excluding site C. So what's really nice about this is that it mitigate the 5' and 3' read bias problem. So I'll go back to the previous plot, which shows the 5' bias. And if I blow this up, you see that most of the reads is actually overlapping the first exon. And in fact, most of the reads are overlapping the N1, which is the non-competitive reads. So let's say if we use the Leafcutter intron usage ratio, the numerator will be I1, but then the denominator will be I1, I2 plus E, but plus N1 and N2, because all these junctions belongs to the same intron cluster that Leafcutter defines. So what this causes is that the N1, because it's just by chance, it's closer to the 5' end, is going to have a lot more reads, and its noise is also going to scale with the number of reads it has. So then the noise would dominate. And we show this using simulation, using like read-based simulation using a real gene. You see that on the x-axis is the predefined ground truth. On the y-axis is the Leafcutter recovered effect sizes. You see that there's almost no correlation between these two because the noise would dominate. Whereas if we use the splice site usage ratio, you see that there's a very nice linear correlation between the simulated ground truth and the recovered effect sizes. So we use simulated data to show that ISSAC is powerful and also well calibrated. So here is QQ plot, sample quantile and theoretical quantile. You see that both ISSAC and Leafcutter are well calibrated as expected. And then ISSAC is also more powerful than Leafcutter across a wide range of effect sizes. So then comes the real data. So here we use the Asian Immune Diversity Atlas data, which we sampled 3 different cell types, from the abundant cell type to medium cell type to rare cell type. So here the CD16 natural killer cell types has about 151,000 cells, which is abundant. And then the gamma delta T cell only has 11,000, which is rare. So you see that for the abundant cell types, we achieve about 40% increase in the number of E genes we map. In the medium cell type, we increase the power by 114%. And then in the rare cell type, ISSAC really shines, where we increase the power by 152%. And this is expected, right? Because in abundant cell types, there's just sufficient power for you to map the sQTLs. But then in rare cell types, because we use metacells, we have more data points to increase the power. And also, ISSAC is significantly faster than the most popular mixed model tool, Glimmer. So across these 3 cell types, we show that ISSAC is 10 to 100-fold faster because we wrote this entire pipeline in C++. And also, we used maybe the last 50 years of statistical innovation to make it extremely fast by not doing matrix inversions and stuff. And to map 1,000 individuals with 1 million cells, it takes about 5 minutes per splice site, and it's highly parallelizable. So if you map genome-wide, it's going to take less than 48 hours. And also, for those of you who want even faster, we're building a GPU version, which the preliminary data shows that it's about 10,000 times faster. So it's going to take about 2 hours instead of 24 hours. Okay, so now we apply ISSAC to dorsolateral prefrontal cortex. So here we used 2 data sets, one from Columbia and one from MIT. And then collectively, they have about 3 million cells over 722 specimens. And then we classify these cells into excitatory neurons, inhibitory neurons, oligodendrocytes, OPCs, endothelial cells, microglia, and astrocytes. So we identified about 29,000 coding cis sQTLs and also 4,000 sQTLs for link RNAs. And you see that most of the QTLs are cell type specific with some amount of sharing across different cell types. So then we asked the question, are these splicing QTLs overlapping with eQTLs or E genes? You see that about half of the S genes are not showing up as E genes, and the other half is showing up overlapping with E genes. So for those that overlap with E genes, we asked, are they sharing actually the genetic regulatory relationship? So we found that 75% of them are actually distinct genetic regulatory relationship, whereas 25% of them share the same causal variant. So then we naturally ask, why would these sQTLs also have the same genetic architecture as eQTLs? So I'm going to do a small digression and talk about intron retention QTL here, and then I'll come back to the shared genetic regulation. So here, in single cell, it's been a challenge to map intron retention QTLs, because in intron retention, these splice junctions are not shared with any other junction, so there's no way to form these intron clusters. So then it's very hard to detect with leafcutter. But we recognize that there's these spliced reads and non-spliced reads. So for splice site usage ratio, it's actually very natural to map the intron retention, right? So we can then use splice site usage ratio to map the intron retention QTLs, and we identified, for example, this intron retention QTL for this gene called NAGK. And it just so happens that it has a H4 of 0.99. We're using colloq here, so the colloquialization probability is very high with an eQTL in the same cell type, which is excitatory neurons. So what we found is that this variant, RS6713, is actually influencing the intron retention of this intron between exon 9 and 10, and it's leading to a premature termination codon, which leads to nonsense-mediated decay, which reduces the transcript abundance. So then going back to the previous point, why are there colloquialization between eQTL and sQTL? We found that actually the majority of eQTL that are explained by sQTL are explained by this nonsense-mediated decay mechanism. So then now the exciting part is that we can actually map the cell state dependent QTL with ISSAC. Right? So here, this is the UMAP of all the major cell types, and here we're focusing on one cell type, which is the inhibitor neuron. It's known that the inhibitor neuron is the most elevated cellular diversity across all neurons, and also we have a sufficient number of cells, which is about 500,000. So we can use principal components to infer their cell state, And so, for example, PC1 is pointing from this sub-inhibitory neuron to this sub-inhibitory neuron. And of course, we can do this for a number of PCs, PC2, PC3, 4, 5, 6, 7, 8. But for the sake of time, I'm going to just focus on PC1. So, we can use the PC loadings to rank the genes that are marker genes for each PCs. So for example, for ARB2, this one has one of the top loadings for PC1. So then we can infer the pathways that are highly enriched for these genes that have top loadings for each PC. Now for PC1, we know that the genes that are overlapping with PC1 loadings is explaining neurogenesis and generation of neurons. So then we can then use this PC loading to map the genotype by cell state QTLs. So here, for example, for this gene PREX2, in this low PC loading space, there is a slightly negative regulation. But in this medium, there is a slight positive regulation. And if you go to the high PC loadings, then there is a highly positive QTL. So in total, we identified about 370 cis sQTLs to be cell state dependent among the 8 PCs in the major cell types. And also, of course, we can detect Alzheimer's disease, which is about half of the population that we have, times genotypes. So here, I'm giving you one example, which is the MT1X. And what's interesting is that this gene is also cell type specific. So it's specific to the astrocytes, which is one of the less abundant cell types, which means that we're not detecting this QTL just because it's more abundant. It's actually less abundant, but it's actually cell type specific. So we're able to detect about a total of 480 bias sQTLs in about 200 S genes. And then, last but not least, we link this to disease by performing colocalization across a number of diseases, including Alzheimer's disease, Parkinson's, ALS, neuroticism, schizophrenia, and Lewy body dementia. And we identified about 350 colocalizations. So here, I'm just going to give you one example, which is TRPT1. So this gene colocalizes between neuroticism, GWAS, and all the 5 major cell types. So we can actually validate this, the genetic effect. So here you see that we have this variant RS115, which is slightly distant from the splice site, right? And it's influencing the splicing level of exon 7. So here we can actually clone out TRPT1 exon 6, 7, and 8 into what we call mini-gene construct. And we created 2 identical copies of the mini-gene construct, differing by only one variant, which is this RS154 variant. So you see that the G allele would lead to exon inclusion, whereas the A allele would lead to exon exclusion, right? Which might change the risk of neuroticism. So just to summarize, ISSAC is a powerful single-cell sQTL mapping method that scales to millions of cells, which I think is important for usage. And also, ISSAC is able to map cell state-dependent sQTLs and intron retention sQTLs, which is not previously possible with pseudobulk methods such as leafcutter. And also, we identified gene by AD interactions and potential risk genes for neurological disorders. And finally, we validated the genetic effects of TRPT1 sQTL. So I'd like to acknowledge our collaborators from NUS, from Rush University in Chicago, from NTU, and then from Columbia, and also our funding sources. And also, I just want to give a shout out to 2 of the very talented students who are presenting posters. They're actually doing clipping on microphones and also running the mics. So you've seen them around you. Wenjing is presenting this evening, and then Liu Fei is presenting tomorrow on Friday. And also, I just want to give a shout out to the FunGene AD Consortium, which supplied us with the data set. That's led by Wang Gao, who's in the audience. So besides this paper, there's already 6 other papers under review, and there's also 6 more papers to be submitted. And if you're interested in our manuscript, which is under review, we uploaded to Mega Archive, but it's a bit slow, so it's in the process of being processed. But I uploaded this to my website, so it's boxianliulab.com/s/isaac.pdf. But the code is publicly available, so feel free to use the code while you're waiting for the manuscript. Thank you very much, and I'll take any questions.
Question on the data generation technology side of things.
Yeah.
Can you comment how much easier or harder would it be for these techniques if the sequencing wasn't 5' or 3' enriched, if it was probe-based, like 10x flex technologies, where you could have specific probes hitting specific introns? Would that be easier, harder? Would love to hear your thoughts.
Yeah. So fantastic question. So the probes, we have looked at the probe list. They don't overlap splice junctions for a very good reason, because they want the probes to be in conserved regions, and in fact, as much conserved as possible. So there are like a very, very small number of probes that actually overlap splice junctions, but if we use flax assays, then it won't give us splicing information, unfortunately. Yeah. Oh, that would be fun. Please give me your phone number. Yeah, like let's have lunch. Yeah.
Okay, one more question.
When you create the metacell, are you able to detect somatic mutations within those reads as well as the splice sites?
You're talking about somatic mutations on the RNA level,
Yes.
Okay. That's a great question. So it's not actually in this paper, but we did have another paper under review, which is called IOLR. In the IOLR, we were able to detect somatic mutations using single-cell RNA-seq read data. It's not covered in this talk, unfortunately, but I will be happy to talk during lunch if you're interested in our other studies. Yeah.
All right, thank you, Boxiang. And that concludes our session. So enjoy lunch.